half minute just to let people finish typing their questions. Otherwise, we'll move on. I think we're going to move on in just a sec. I just want to mention that Gregory was saying that this seems to be a very intriguing line of work. First saw it at NeuroHike. Oh, cool. And very much look forward to following the progression. Yeah, I feel like we're pretty lucky to be able to have the Neuromatch Conference. Otherwise, we would just all be sitting in a vacuum. But yeah, it's definitely good to be able to see these things progress. All right, so I think we'll go next up to Carolin Ruven at the Burke Medical Research Institute. She'll be talking about microsurgical lesions identify segmentally distinct responses of axotomized corticospinal neurons. All right, go ahead, Carolin.
Okay. Okay, hello everyone. So thank you for giving me the chance to talk about my work. So I'm Carolin Ruven. I'm also from SANI Lab in Burke Neurological Institute. And I'm gonna talk a little bit about how I'm using microsurgical lesions to study corticospinal tract regeneration in development. So corticospinal tract, as already previously introduced a little bit, so it's formed by the neurons in the cortex and their axons that extend to the spinal cord. When spinal cord injury happens in adulthood, then this tract is not able to regenerate. So since this tract is needed for voluntary motor control, the regeneration of corticospinal tract remains a key focus for recovery after spinal cord injury. So it does remain well established that intrinsic regenerative ability of neurons declines with age. So there has been extensive research on identifying what's behind this failure in the adulthood. So they found out that in adulthood, the combination of extrinsic factors and intrinsic factors are responsible for the regeneration failure. So extrinsic factors are at the injury site and are mainly inhibitory for axonal growth. These are, for example, glial scar formation, inflammation, cell death, inhibitory proteoglycans, and myelin-associated inhibitors. These are known, most of these processes are known to become more prevalent with age. So at the corticospinal neuron level, there are different growth-associated genes and transcriptional regulators that control the axon regeneration. So in adulthood, these developmental growth genes are down-regulated. So despite the significant work, we still don't really know how and when actually in this developmental period, this regeneration ability gets lost. So it's also because the current models that are there for neonatal spinal cord injury are very severe and cause massive tissue damage. So in this, so for example, this is one of the conventionally used model, that's spinal cord injury compression in neonates. And as you can see already from the whole mount images, the tissue damage is very severe. And because of that, it's difficult, you're not able to distinguish whether the axons are not able to grow because of they have lost the intrinsic ability to grow, or it's because of these extrinsic factors at the injury site. So therefore, to be able to actually answer the question when and how does the regenerative ability get lost, we needed to firstly come up with a new approach that would leave the spinal environment relatively intact while still precisely disrupting the CST. So for that, we actually established a novel microsurgical approach to transect the developing CST in the neonatal spinal cord. So in our model, we are using a beveled micropipette that here you can see inserted into the XV1 neonatal spinal cord for the size comparison. So this pipette is then further attached to a high-frequency vibrating device, and the injury is done under ultrasound-guided backscatter microscopy. So here's just a short video clip showing how, the injury process. So to firstly orient you, The animal here, the mouse pup, is on the side. With the dotted line, you can see the skin. Here in white, you see this the spinal vertebrae, and this here is the spinal cord and the corticospinal tract. So in the red here will be the micropipette. So here you'll see the micropipette inserted into the spinal cord, and then the vibration is turned on to cause the injury. And the wavy movement is just the artifact from the ultrasound. So as I showed before, that conventional models result in this massive tissue disruption that you can already see in whole mouth. Then in contrast, after our microsurgical injury, there's barely any visible damage to the spinal cord. And overall, it also results in minimal glial scarring, inflammation, and it leaves the spinal environment largely unperturbed while precisely still guiding the CST. So using this microsurgical injury approach, we studied the corticospinal regenerative ability at different spinal segments at the neonatal ages when the CST tract is still growing. So firstly, we did the injury at cervical C2 level at B4 time point. So what we found there that this CST and axons totally failed to grow across the lesion site at this age. So then we also went to the injury at the same time point at B4. We injured the axons at D11, thoracic D11 level segment, where then the injury site is closer to the growing CST tips. And what we saw is, in contrast to the previous one, that the axons are actually able to grow through the injury site. So next, we actually went a little further in the development to the P7 age, when most of the tract has grown further. And we saw again that actually the tract is still able to grow through the injury site. And in this T11 segment, actually, we saw this regenerative ability. We started seeing the loss of it at P14. So here's also just to quantify this by showing the amount of axons that make it below the lesion site to the lumbar cord from the cross sections, and you can see clearly that there are no axons making to the lumbar cord after the cervical injury down there after P4 and P7 low thoracic injury, and the decline starts at P14 for a low thoracic injury. So then the main conclusion basically that I wanted to convey today here is that the regenerative ability of CST is lost earlier than previously thought. It's already lost at least by P4 in cervical segment, and the loss of this ability is not uniform across the entire length of the axon. So next, we do actually have some mechanisms that we want to study that might be behind this phenomenon that we are seeing. So to kind of maybe facilitate the questions in the discussion session, I wanted to just bring up some of the data that we're gonna study next. So first, we do think that it might be because of some CSN intrinsic factors. So for example, there might have been a developmental switch in a growth program after the axons have passed the cervical cord. So as previously maybe mentioned already by Julia, that there are different molecular programs kind of targeting the axons either the cervical or thoracolumbar cord. So if in the case that the growth program has already turned from cervical to thoracic growth, then maybe that's why after the injured cervical, the axons don't know how to grow anymore. Other option might be that also after the injury, there is just different injury response that gets activated depending on the injury level. Also, it might be still, we don't exclude that it's somehow CSN extrinsic environment, even though in our model, we try to minimize the effect to the environment. And we don't see much of the glial scarring and inflammation, but still it is possible that either regionally distinct astrocytes, myelination, or maybe microglia can somehow affect the CSN axon growth differently. So before we get to the questions, also quick acknowledgement for my PI Vibusani and all the lab members, our collaborators, and also our funding. So now I can leave this on for maybe facilitating some of the discussion.
That's an excellent idea. I appreciate this slide. Thank you for that excellent talk. All of these have been really great talks, you guys. I just wanted to mention a comment from Kelvin Jones, just saying that this looks like a really nicely controlled injury. So great job there.
Thank you.
All right. So we will wait again for people to send in their questions via the Q&A box or through the chat. And as usual, feel free to raise your hand and we'll bring you on. We can make this segment be specific for Carolin. And then after a little bit, I'll just open up to questions for any of the panelists, or people can just come in and have a conversation with the panelists. All right, so we have a question from Kelvin Jones. Where was the injury relative to the transition zones, the Julia's transition zones?
So the cervical injury, the higher one at C2, so it's below the brainstem cervical transition zone, but it's before the next cervical thoracic transition zone. So yeah, so at that point we should be injuring the axons that were heading to cervical or thoracolumbar, all of them.
It's also maybe worth noting, highlighting Carolin's work again, that she did the same experiments also with a marker only looking at the thoraco projecting neurons, and she sees the same issues. So it's not a subpopulation question, but the question of where the axons are actually transected.
Yes, exactly.
That's great to know. Thank you both. Thank you for clarifying that. All right, we have a question from Gregory, who's just saying, excuse some ignorance about this area. Don't worry, I feel similarly. But I am curious if there's any way to assess differences between ascending and descending tracts in their ability to cross the injury site.
We have not looked at the ascending tracts, but there are stainings that could be done for the sensory tracts to also see if they cross the lesion site or not. So in our model, we don't. We are saying that we're looking at CSD and we're damaging mainly CSD, but we are probably still damaging some ascending tracts, the same dorsal funiculus. So we have not looked at if they are able to grow or not. But this is something we definitely are actually planning to look at.
Yeah, that would be very interesting. Another question from Kelvin. If you injure at that level but have cervical CSN express CRIM1, do they grow past the injury?
I don't think this is an experiment that has been done.
Yes, we have not done. We have done the opposite way. No, we've actually. So we have done, not CRIM1, one that is somehow similar, one of the molecules that neurons, and we have actually tried to overexpress this after our T11 injury. And at the time points at P14, when we see already that they don't grow normally. And after we've overexpressed the cerebellum one, we have, our data is still not very conclusive. I don't think there is a big effect, but that was our idea. Maybe it will be a combination of different genes to get them there. But at the moment, it seems it doesn't actually make it. But we haven't tried the CRIM1.
But to also add to that, we don't think that CRIM1 is necessarily involved in the regeneration capacity of the developmental neurons. So I personally wouldn't expect any effect on if we overexpress CRIM1 in the cervical projecting population and then injure.
Yeah. Because also, I mean, as said, the population that I was showing now that we injure at the cervical level, it is mixed population of the ones that go to cervical or thoracolumbar, but the growth stops for all of them. So even if you try to make cervical to go longer, at that level, it was even stopping the long growing ones. So in that sense, also, it's unlikely that it will make a difference for that. But good question.
Yes, thank you. That was a really good question, and your responses were very illuminating. Thank you both very much. In general, thank you all for your questions. They've been